is right. I don't know if this thing will come on. There we go. Okay. 1/3 of the people tested were not able to detect sweat odors. We're talking about 18,000 people that did this survey. 1/3 of them couldn't smell sweat, and 30% couldn't smell musk. And musk are, of course, sexual odors, similar to sexual odors, even in humans. So we're talking about a relatively large percentage of the population. If you consider 1 out of every 3 people can't smell select odors. That's a lot of people. So that's enough people so that when we start talking about pheromones, that we have individuals that can't smell it. Odorants enter the nasal cavity in the form of free-floating chemical molecules in the air during inhalation. So if it can't break down as a chemical, you can't smell it. And this is a problem with natural gas. Natural gas is poison, and we can't smell it. So in order for us not to be poisoned by natural gas on a continual basis, they put sulfur in it to give it an odor, and it smells like rotten eggs. Well, of course it smells like rotten eggs, it smells like sulfur. And of course that's why we can detect it. Otherwise it would be odorless, and we wouldn't be able to detect it. But we can, of course, since they put the sulfur in it. The odorants first encounter the mucosal layer, which binds the chemical to a protein for transport to the olfactory epithelium. You can see how slow this process is. The viscosity of the mucus layer determines how rapidly an odorant reaches the cilia of the olfactory receptor. In other words, if you have a dry nose, if you're out in the desert and you have no mucus, then you can't smell a damn thing. If you have a cold, you can't smell a damn thing. Who was I talking to last night? He's got a sign. Oh, it was my next door neighbor, Ken Losey. He teaches biology and chemistry, I think, over in the science wing. Anyway, he's got a sinus infection. He can't smell anything. And he talks funny because his sinuses are full of mucus. The odorant then reacts with the receptor proteins located on the surface of the olfactory cilia and dendritic knob of the receptor cells. These proteins are members of a complex family of proteins referred to as G-protein-linked receptors. You can see how complex this is. The G-protein-linked receptor combines with a molecule of GTP, which displaces GDP. The G-protein alpha subunit dissociates and activates adenyl cyclase, which produces cyclic AMP. So you can see how slow this process is. So smelling things is the slowest process we have, the slowest sense that we have. Cyclic AMP binds the sodium channel and opens it. The sodium enters the cell. This creates a generator potential, which is interpreted in the brain as a specific odor. The receptor protein returns to an unbound state. And there we go. That's how smelling takes place. And you can see how slow the whole process is. Pheromones. So we're going to talk about pheromones. And there's a reason why I believe in pheromones. I've told you some of my stories. I told you about the Japanese stewardesses. Well, let me start the story all over again. I don't know if you've heard the whole thing. My wife was stationed in Korea. The assignments to Korea are isolated tours, which means she had to go over there all by herself. Well, it was a 13-month combat tour to Korea, even though there wasn't any shooting taking place. Well, not a whole lot of shooting taking place. Anyways, a 13-month combat tour, and I hadn't seen her in 6 months. So I was going to fly over there. This is in October, wasn't it? No, it was in August. Anyway, I was going to fly over there and spend a couple of weeks with her. And I hadn't seen her in 6 months. And geez, how old was I at the time? Let's see. That was 1994. In 1994, I was 55. No, I was 45. I was 45 years old. So I was still a fairly young person. I was still a pup. Okay, I was still pretty young. I was young, she was young, and we were both sexually active individuals. So 45, my goodness gracious, I was only 45 years old. So, and I hadn't seen her in 6 months. So I was really, really, really ready to see her at age 45. Anyway, so I jumped on the airplane, and I was in Oklahoma City at the time. So I jumped on an airplane in Oklahoma City, and I flew out to Los Angeles. And then we got on a flight to Japan. When they fly to Japan, they don't fly across the Pacific. It's too dangerous. What they do is they fly around the Pacific Rim. So we flew up through Canada, up along the Canadian coast and up along the Alaskan coast and then down into Japan. And that's how we flew. It was an 18-hour flight, and it was a jumbo jet. And it was a JAL flight, Japanese Airlines. Japanese are very polite people. A lot of things happen on the flight. I think I got up and went to the bathroom once. Anyway, and I read most of the time, and I slept part of the time. They had 2 meals you could have. You could have chicken or you could have fish. And by the time they got up to me, I was up in the top part. So I don't know if you've ever been on a jumbo jet. It's got the normal place where everybody sits, and it's, I don't know, there's like 500 people down there. Then they've got an extra 200 seating that goes up. That's up here. This is where the pilots are. Okay, so what they did, they fed everybody down below, and then they fed everybody up above. Well, I'm allergic to fish. And by the time they got upstairs, they'd run out of chicken. So I told them I can't eat fish. I'm allergic to fish. And they got all upset. And they found me some chicken somewhere, where I don't know where they found it. Anyway, they found me a chicken dinner, and it was a good dinner. That wasn't the problem. And of course, before dinner, they give you a warm washcloth to wash your face, and after dinner, they give you a warm washcloth to wash your face. So all this is taking place, and these stewardesses are just sprinting up and down the aisle. I mean, they're just really booking it all over the place. And there were just a ton of them. There must have been 15 or 20 of them. But then again, there's 700 people on this flight. So we landed in Narita Airport in Tokyo. And at Narita, you land way out here. You land way out here, and it's like a mile. You land at the end of the runway. You land at the terminal, and then they fly out to the end of the terminal, and then they transport you. They put you on a monorail. Okay, so they put us on a monorail. Well. And of course, the people that got off first were the ones that didn't need to go through customs, and almost everybody on the flight was Japanese, so they all had to go through customs, and I didn't. So we walked through this micro terminal. It's not the real terminal. The terminal's over here, but that's where all the customs were. So all these guys, almost everybody that got off the plane had to go through customs, except me. I was flying on to Korea. So I got off the plane, walked onto the monorail, and I was like the first one I was the first one there. So here I am. I'm kind of sitting in the corner. And the next people that got on were all the Japanese stewardesses. And if you've ever been to Japan, of course, the Japanese are it's always crowded. Everything's crowded in Japan. So I've got all these Japanese stewardesses. I got them everywhere. I got them in front of me. I got them in back of me. I got them to the left. I got them to the right. I had Japanese stewardesses surrounding me. And they're just jabbering like crazy. And of course, they're the little doll people. They're so cute. Anyway, okay, so I'm surrounded by Japanese stewardesses, and it was okay in the beginning because they were, they were a step away from me, and then all of a sudden there was this huge surge of people, and they just packed in as tight as sardines. So here I am standing there, and I got Japanese breasts pressed up against every part of my body. This is so weird. And I don't know why the lady in front of me turned around. But she was facing me, and she was talking, I guess she was talking to the lady behind me. Of course, I'm an American, so I'm 5'6", and these Japanese women are like 5'2", and so she's kind of going up on her tiptoes to talk to this lady. It's really kind of interesting. So I got a lady in front of me, I got a lady behind me who she's talking to, and I've got women all around me. And so then the train starts. It's like a standing lap dance. It was so weird. And these ladies, of course, they've been sprinting for the last 18 hours. So they should be, nice and odoriferous. They weren't. They used some kind of corn, I don't know. And they don't really hardly sweat at all. Yeah. But it smelled like corn, is what it smelled like. They all smelled like corn. So it's cornstarch is what they use. Okay. Anyway, so here they are. I'm getting this standing lap dance. And I'm not reacting to it at all. At all. I mean, it's like I'm not a male at all. I got all these women rubbing their body parts against me. And I'm telling you, they were rubbing against every place I pushed out, my body. I've got some lady rubbing up against me. I mean, it was just really strange. And I'm not reacting at all. Not at all. And of course, I'm starting to worry. I mean, and it was a mile. It was a mile from here to there, and it must have gone 25 miles an hour. It wasn't going very fast, but it was rocking from side to side. So I've got women rubbing their chest against me, like, it was interesting. It was very interesting. And the whole time they're just jabbering away, and this lady in front of me, I mean, she gets really close, and I can smell her breath to the extent that I could smell her breath. Luckily, I had put a piece of gum in my mouth, and I didn't have bad breath. But this lady, I could smell her breath. I mean, sometimes when I kiss my wife, I don't get that close to somebody. But here she is. I mean, she's literally up in my face. And there's such cute little things. They're very attractive, Japanese women. And the JAL only hires young, attractive females. So anyway, so I'm getting a standing lap dance. And I'm not responding at all. I mean, it was spooky. So that night, I'm laying in bed, waiting for the morning so that I can get on the plane and fly to Korea. And I'm thinking, this isn't going to be fun. I mean, I've been anticipating going and visiting my wife, and I'm only 45 years old, and here I've lost my ability to function. So I was scared. I was more than a little scared. So I got on the plane the next day, and of course everybody on the plane were Korean. Well, most of them. Some of them were Japanese, and there was me. So we get off the plane, and this time I had to go through customs, along with everybody else. Everybody had to go through customs. So I finally got out, and there's my wife. She's standing there with a 6-pack of Mountain Dew. It wasn't the Mountain Dew, okay? So as soon as I saw her, I ran up to her and gave her a really big kiss, and at that point I realized everything's going to be fine. Everything's going to be fine. Of course, the Koreans and the Japanese don't touch each other. They bow to each other when they say, they don't hug, they don't do. And here we are, we're in this lip lock, and of course they were seeing it as a pornographic movie, I'm sure. Anyway, and I was there for 2 weeks, and it was like a second honeymoon. I mean, we had a great time. But then thinking back, I'm going, well, why in the world didn't I respond to these skinny little cute little doll-faced Japanese women. I mean, that doesn't make any sense. And then the answer was that Americans, people of European descent put off pheromones. And of course, the Japanese don't. And the reason Japanese don't is because they've had arranged marriages for hundreds of years. So they've lost pheromones. They've lost all their pheromones, which I really kind of wonder how Americans can go over there and marry a Japanese woman, because I mean, they have no odor, and odors are really important. I mean, they're important to dogs, they're important to cats, they're important to, they're important to Europeans. Maybe Europeans are just there's something wrong with them. Anyway, so pheromones do exist for me. I know they exist for me. Here I had not access to all these women, but I mean, it was kind of an interesting situation. But I was getting no orders off these ladies at all. Nothing. It was like, I don't know, just standing out in the middle of the wind. I mean, it was that bad. Another time, this is when I was in Mississippi. I went to get a Little Caesars pizza, and as soon as I stepped in the pizza parlor, I could smell it. I mean, there was something going on. It wasn't a strong odor, but I mean, it was almost like it was raw sex. There wasn't anything happening. It was just that the lady behind the counter put off pheromones. Why she put off pheromones? Maybe her boyfriend had just come in, and at that point you put off pheromones, and maybe that's what had happened, or maybe she was flirting with the guy in the back. I have no idea. But she put off pheromones, and it was so strong that it lingered with the pizza. So I paid for my pizza. I went outside, and it was still there. It was on the pizza box. There was something. There was something, and it took. It was about a 20-minute drive, and it lasted for about 15 minutes. I'm going. I don't know what I'm going to do. I'm about to go home and feed this pizza to my wife. What's going to happen next? But after about 15 minutes, it dissipated enough that I didn't have any reaction. So I know they exist. They do for me. I reacted, and the little girl behind the counter was probably, 16 or 17 years old, maybe 18 years old. It's hard to tell. She was African American, but she had the oddest yellow eyes. Yellow eyes are kind of a light brown, but her eyes were almost yellow. I mean, it was really kind of interesting. And it wasn't that I was attracted to her face. I don't even remember what she looked like except for her eyes. I wouldn't remember anything except that she was African American. So here she is, she's putting off these pheromones, and I'm reacting to it. If another guy had come in, he probably wouldn't have reacted to her pheromones. Because the connection between people, there's about 150 different configurations of pheromones. We either react or we don't react. Most people don't put it off those odors that you would react to, but every once in a while you'll run into somebody who puts off the right pheromones. Anyway, they did experiments with this stuff in Switzerland, and they took men's t-shirts and they put them in plastic bags, and they had a group of women smelling the dirty t-shirts. And in most cases, they would go, this stinks, this stinks, this stinks. And every once in a while, they'd come across somebody who they thought smelled sexy. It smelled sexy. It was really kind of interesting. They did the same study in Philadelphia and got exactly the same results. So I don't know. You may be one of the 1/3 that doesn't react to body odors. You may be one of the 30% that can't smell sexual odors. These people exist. The Koreans eat too much garlic, and it masks their odors. Japanese don't put off any pheromones at all. So it should be interesting. I mean, the Japanese don't have arranged marriages anymore. You just wonder, and they seem to be they're at zero population growth, so they're not actually reproducing at the right level to maintain themselves.
I could remember when I was a little bit younger. I used to work for a moving company, and we would move people's stuff from high store buildings out. Anyway, I remember you go into their homes and you can just smell whatever.
They're the way that they smell.
Yeah, it's like it could be anything.
It might be the grease that they use to cook their food with.
Yes, that one. Or it could be just like linen, or it could be just like a sweat.
Sure.
Even just like, if you get there early in the morning, you can smell, I think, what they breathe inside the room the whole night.
Right.
If it's Oriental, it'd be like a garlic or something. I don't know. It was like some kind of like.
If it was Indian, it was curry.
Yeah, I get that out. That's true.
Well, we smell like what we eat anyway.
Yeah.
I mean, even your urine. I eat a lot of cauliflower and cabbage, crucifers, and I can smell it in my urine. Most people don't smell urine, but I've had a lot of experiences. I used to do urinalysis ad nauseum, and literally ad nauseum. So I'm pretty sensitive to smells. I used to be able to detect if somebody was pregnant from their urine. It was weird. Yeah, because they put off a really interesting, a strange odor. It's a sexual odor, but of course she's pregnant, so it's changed her hormonal balance, and that's what you're smelling. As odd as that may seem, most people don't come in contact with urine that much, especially other people's urine, unless you work in the lab. So, anyway, so pheromones are detected through the vomeronasal organ. The vomeronasal organ is actually lower in the nose than the olfactory bulb. The olfactory epithelium is way up in the top, and the vomeronasal organ is much lower in the nose. So you detect sexual odors before you detect smells, as strange as that may seem. And you may react to those sexual odors before you even can detect what that odor is. While researchers have thought that the vomeronasal organ is vestigial in humans and other high primates, the organ still exists, and some people report responding to it. And like I just did respond, I just did report responding to it. So I believe that it's there. Now this is a video about pheromones.
Well, are you looking for love and failing to find it? In the past, retailers have sold love potions that claim to attract the opposite sex, even though none of it's been proven to work. So we were curious about the new batch of potions with the secret ingredient pheromones. They're in some of the new Christmas fragrances, but do they work? Bill Bitter did a little experiment to see if they're too good to be true.
I think our product is the only product that has any proof behind its claims.
Biologist Winifred Cutler is perhaps the nation's leading pheromone champion. She claims her pheromones work for 75% of the people who use them. The price for 1/6 of an ounce of her pheromones, sold through a company she calls the Athena Institute, is $100.
Our pheromones are sexual attractants. They do make the wearers more sexually attractive. That's very clear.
Oh yeah, I know what you're thinking. Why would attractive 20-somethings even need pheromones? After all, it's those of us who are no longer in our 20s who could probably use a little help.
Women in their 40s are not excreting the same level of pheromones they might have been when they were in their 20s or their 30s. And for those women, adding the pheromone to their perfume can be just the kick that restores some of what they thought they had lost.
So to see if pheromones work on middle-aged women, we enlisted the help of Kathleen and Lisa Ann, Both over 40, both still single. Lisa Ann is a newspaper reporter, and she was excited to find out if this would work.
I do get attention, and I do have some dates, but I do not have a committed relationship, which I would like to have.
Kathleen calls herself New York's Lady Barber. She says she's never had any trouble meeting men, but
I'm one of 8 kids, and when I go home to my families for Christmas, everyone's married with kids except me. I just would love to have a boyfriend for Christmas this year. Now see, I'm gonna cry. This is so pathetic.
so Kathleen and Lisa Ann agreed to wear the pheromones every day and keep a video diary.
Hi, this is Kathleen Giordano reporting for Pheromone Duty. I'm getting lots more attention on the street, so I have to assume that the pheromones are working.
I'm wondering if I'm seeing any changes. Only subtle so far. Don't have men falling over me yet.
Crossing 2nd Avenue this evening. 3 guys, 3 friends, walking abreast crossing the avenue said, "Oh my God, you are so beautiful."
I'm still not noticing a whole lot of action.
I didn't think that too.
Yes, okay, good.
Lisa Ann was trying. She even learned tango, maybe the sexiest dance ever invented, but she stayed mostly on the sidelines. 1 month into the experiment, Kathleen and Lisa Ann got together to compare notes. Kathleen was dating 3 to 4 times a week, more than ever.
He said, "What are you doing Saturday?" I said, "I have no plans." He said, "You do now."
Oh, yeah!
So the work part of my life is good. The dating part of my life can use some improvement.
We'll check back with Lisa Ann and Kathleen after another month has passed.
Huh. Is that it?
Coca-Cola doesn't say what's in its formula. Athena does not either.
We check back with our 40-something women. Kathleen still didn't have a Christmas date, but she remained convinced that pheromones were working.
I think the pheromones are like that added little secret a woman keeps in her arsenal.
And as for Lisa Ann, something of a surprise, she's become a pheromone convert.
It took a really long time, and I was really beginning to suspect it was hooey, but something started to happen. There was a transition.
On a cold December night, Lisa Ann and her date keep each other warm. And guess what? She's already decided what to buy herself for Christmas. A new supply of pheromones.
That was interesting. So who was reacting to it? In that case, it was...
We're all attracted to people all the...
Yeah. So the black lady was having problems attracting black men. But if you noticed, her boyfriend was white. So this may be a white people thing. When we're talking about 75% of the population, when we're talking about 70% of the population, that's the number of white people in the United States. So it may just, this pheromones may just be for white people, as odd as that may seem. I mean, the white lady, everybody started reacting to her right away. When the black lady, people weren't the black males weren't reacting to her, but white males were. So this may be a white thing, as odd as that sounds. Let's see if this works. Here we go.
You Have Two Noses by Mike, Sidney, and Will. Some scientists argue that the human nose is becoming obsolete. As distressing as it is to imagine a world where glasses are now impossible to wear and Eskimo kisses are non-existent, could there be some truth to this claim? From an evolutionary standpoint, the human sense of smell has been pushed aside in favor of more primary senses, such as sight and hearing. We treat the sense of smell as base, since it is more useful for animals than it is for humans. But is this an accurate claim? In the scientific community, the existence of human pheromones is a hotly debated topic, even though their presence in other animals is widely documented. If humans do have pheromones, it could completely change the way we think about our olfactory system and possibly help explain some of the mysteries of human interaction. There are multiple forms of pheromones that we will be discussing. Dispersal pheromones are typically used to scatter a group. Alarm pheromones are used to warn other members of the species to a danger. Aggregate pheromones are perhaps the most famous and widely studied. They include the famous attraction or sex pheromone as well as chemicals used to lead members of their species to food. To begin, we will primarily be looking at dispersal and aggregation pheromones in animals, and then move on to critically analyze whether or not humans do indeed possess this extra sense. One example of dispersal pheromones occurs in certain kinds of ants. A chemical is released by dead ants that alerts the rest of the colony to transport the corpse to a cemetery of sorts. When scientists isolated this chemical and covered a live ant with it, the colony proceeded to bring it to the cemetery, despite it not being dead. When the ant escaped, they continued to drag him back to the cemetery until the chemical wore off.
It's cruel.
One type of aggregation pheromone is the ever-popular sex pheromone. Female silkworm moths release a chemical called bombakill, which is dispersed by the wind and can attract males from several miles. When scientists placed a female moth in a glass jar, preventing the chemical from reaching the male but not obstructing view, the male had no attraction. However, when the female was placed in a mesh enclosure, out of the line of sight but allowing the chemical to be released, the male was still attracted to the female, despite being unable to see her. This proves that vision is not the driving factor in the attraction of silkworm moths to each other. The antennae of the silkworm moth are incredibly sensitive to this chemical, able to detect as little as 1 molecule of bombakill. Interestingly enough, the male antennae are considerably larger and more sensitive than the female ones. Pheromones have also affected the behavior of mammalian species that are more closely related to humans. Examining these will allow us to deeper understand our own possible connection to pheromones. For example, consider the mouse. Studies have been done that show if mice are deprived of their olfactory senses before being sexually active, they will never exhibit sexual behavior. This cannot be a coincidence. There must be some kind of connection between the nose and the reproductive system. The connection is slightly convoluted, but it goes through the brain, like most things. The ovaries and testes contain endocrine glands that secrete sex hormones. In females, the release of hormones is cyclical and determines the timing of her estrous cycle, or the release of an unfertilized egg and when she goes into heat. Controlling the endocrine glands is another gland, the pituitary gland. It's located here, at the base of the brain. It releases its own hormones into the bloodstream, which tell the endocrine gland when to release its hormones. A structure called the hypothalamus, located here above the pituitary gland, in turn controls the pituitary gland. It also releases hormones. These tell the pituitary gland when it needs to release its hormones. The hypothalamus gets its inputs from a variety of places, one of which is the endocrine glands themselves. When the endocrine glands have released enough sex hormones, the hypothalamus gets a signal to stop sending signals to the pituitary gland to send signals to the endocrine glands. Very circular. Another system that talks to the hypothalamus is the olfactory system itself. In order to understand how this happens, we must look at how the nose is built. The inside of the nose is lined with 3 kinds of cells. One has to do with breathing and nothing to do with smell, so we'll ignore it in this presentation. The other 2 are the olfactory epithelium and the vomeronasal organ. The olfactory epithelium is located up here, at the bridge of the nose. This is the system that allows what we think of as smell. The smell of chocolate, cologne, and the rancid stink of a full diaper, all through the olfactory epithelium. The receptors in this area transduce odorants into electrical charges, which then go to the main olfactory bulb, and then the hypothalamus. This is the vomeronasal organ, located in each nostril. It looks and operates very much like the olfactory epithelium, but has some marked differences that encourage scientists to think its purpose is distinct. One notable difference is the physical distance from the olfactory epithelium. Another is that the brain keeps their offshoots separate. The vomeronasal organ sends information to the accessory olfactory bulb, rather than the main olfactory bulb, before going to the hypothalamus. The brain is very rarely arbitrary, so this separation must mean something. The separation occurs in all mammals, but so far scientists have really only been able to prove anything in lower mammals. One of the few well-documented cases of pheromones in humans is the synchronization of menstrual cycles in women. Most women will swear up and down that this happens, but one of the first studies that proved this occurrence was done in 1971 by M.K. McClintock. She had a group of incoming freshman women assigned to one dorm report the timing of their last menstrual cycle. Even though their cycles were at different times initially, they were synchronized within 4 months. McClintock went on to do further studies about why the synchronization happens. A study performed in 1998 by McClintock and Kay Stern took odorants from women's armpits and exposed them to other women's vomeronasal organs. The synchronization of cycles occurred even though there was never any social interaction. McClintock repeated the study using an odorant from one male donor. The variability between cycles decreased with this experiment as well, but not to the point of the previous study using female odorant. As if women weren't affected enough by these suspected pheromones, an additional study done this year by psychologists at the University of Padova in Italy investigated the role of chemosignals on a female's rating of male mates. The experiment used either androstenedione, the most intensively studied steroid and possible pheromone, or a different control compound on heterosexual women. The women in both groups were then asked to view male and female faces while their eye movements were recorded. The results were that females at high conception risk spent more time looking at the female faces, regardless of the compound administered, and those at low conception risk only preferred female faces following exposure to the androstenedione. In essence, when reproduction is critical, there appears to be more of an evaluation of potential competitors. But when conception is unlikely, odorants or possible pheromones such as androstenedione might incite the same competitive response. As appealing as the idea of a bottle of pheromone-infused cologne may be to some people, scientists still have a long way to go before they fully understand how pheromones work in humans, or if they even exist at all. As we've discussed, the existence of human pheromones is a hotly debated topic in the scientific community. However, we believe that there is some serious biological evidence to suggest that, at the very least, pheromones were once a vital component of human interaction, and perhaps may still play a role. Obviously, this topic requires much more research and study before scientists can even come close to fully understanding the role of pheromones in humans, but is certainly an attainable goal. After all, the scientist certainly knows what they are doing.
That was funny. That was clever. All right. There we go. One last one. Brainiacs. This is out of England.
Have you ever seen a real ugly bloke walking around with a gorgeous woman and thought, how? Well, is this the answer? Pheromone spray, available from your local sex shop. Now, it claims to make you irresistible, even if you're a short, fat, ugly bloke with no money and all the personality of an ironing board. But does it work? This is Julie. Julie is lovely. Julie wants a boyfriend. Which man is right for her? We've got a random selection of 3 fellas for her to choose from, all with their own distinctive smells. Which one will she choose for her blind date? Arms up, please. Your face. The time can't erase in mine to my eyes. But the stage just has stopped. So many nights like this. Let's take a look out and see. And stop sniffing, please. I love you so. please. Arms up, please. So, Julie has had a nose full of our three lads. Which will she choose to be her blind date? The pheromone boy, the aftershaver, or will it be Mr. Sweaty?
It's just sweaty.
The results coming up. Back to our blind date. Our lovely lady Julie is ready to choose her new boyfriend just on the basis of his smell from three fellas chosen from a cross-section of society. Knowing that the sex shop pheromone spray, its main constituent is androstenol, a factory-made chemical which mimics men's natural sex hormones. A liberal splashing of it should drive women crazy, but does it? Our other men smell of natural sweat and aftershave. Will they turn her head instead? She's sniffed them all, and now she has to choose her favourite. Please take your blindfold off. So tell me about number one.
What's going to stunk?
Really?
Yeah.
Stunk of what?
BO, I would say BO.
Okay. And tell me about number two.
He's not nice, a lot better than number one.
Yeah? And what about number three?
He was in between. Nice, in some areas, a bit stinky others.
Mediocre.
Yeah.
Okay, who have you chosen to be your blind date for this evening? Number one, number two, or number three?
Well, it's got to be number two, definitely number two.
Okay, so you've chosen number two, so let's meet the two that you've turned down. Come in, Fabio.
Sorry.
Come in, Dal.
What's this?
Hi! Sorry.
And your blind date for this evening is Jimmy from Scunthorpe, and he was wearing aftershave. Come in, Jimmy. So there it is. Cheap aftershave beats pheromones every time. Splash it all over, boys.
That's too funny. Okay. So you can believe in them or not believe in them. It looks to me like watching these and then with all the information we have out there, it may just be a white person thing. I don't know. Anyway, that is the end. There we go. Okay. This is the 10th week, and this is the 10th chapter. So I'm caught up somehow. Chapter 10 deals with vision. I come from a really interesting family. I have really good vision, even good peripheral vision. So if I see movement out here, I'll look to see what I'm looking at. I have a brother with tunnel vision, so his eyes are deep set into his skull, and this is about it for him. So it's really kind of interesting. And I already told you that because of that, he sees better at night. He also has brown eyes, and I have blue eyes. But he has the ability to see very well in the dark. He's also invisible in the dark. I haven't figured that one out yet. We used to play night. We used to play, hide and seek. And pee-wee stick down and all kinds of interesting games, kick the can, and we could never find it at all. He just has the ability to understand or to recognize shadows, to get in the darkest places, and just stay there. And we could never find him. Even with flashlights, we couldn't find him. He was just amazing. The whole area, and of course, I told you what he did. Yeah. The whole area that you can see without moving your head or your eyes is known as your visual field, and I don't know how wide your peripheral vision is. Mine is, so I lifted yesterday, so I could barely go out. I can still see this, So, and that's really kind of amazing if you think about it. While the visual field may be wide, an individual's visual acuity, of course, tends to only be in the middle of the field. So despite the fact that I'm looking at the whiteboard over there, I can still see the chair. I can see you. I can see behind you. I can see way over here. But of course, I couldn't really tell. Once upon a time, I was running on a treadmill. I think I've told you this story. I was running on the treadmill, and I kept seeing a yellow light. What I was actually seeing was a red light, but of course, since I was seeing it in my peripheral vision, I saw it as yellow. And when I looked for it, I couldn't find it. And this may be one of the reasons why we see ghosts and whatnot. All we're doing is detecting light over here, and when we look directly at it, it's not bright enough for us to see. So, but I can see that light, and if I try to catch it, I can't catch it. This is because there's a concentration of cones in the middle of your visual field, and of course those cones make up your fovea. And for that reason, right in the middle of your visual field, you have the best visual acuity. However, even with the concentration of visual receptors in the center of the visual field, there is a spot where the optic nerve feeds out of the eye, the blood vessels feed into the eye, and this is known as your blind spot. There is actually no light detectors in that portion of your eye. Normally the brain automatically fills in the blind spot, so despite the fact that it's there, you do not recognize it. Vision comes to humans in the form of wavelengths of light or photons. The human visual ability is somewhere between 400 and 700 nanometers. And it's what we refer to as ultraviolet to infrared light. Other animals have ranges of vision that encompass wider spectrums of light. Bees can detect beyond ultraviolet light, and that's one of the reasons why they can follow a trail from another bee. So all a bee has to do is dance and tell them, I found flowers, and they'll be able to follow the bee's trail back to the flowers. Because he has put off this ultraviolet signature, and they will be able to see.
There's something I want to mention about vision. I have a friend who has his vision started dissipating as he got older.
Yeah.
I think from junior high.
Right.
And he's probably in his late 30s, but it's closed up now to where he can probably only see like a pinhole through a paper. But during that time, when he was losing all his vision, he could pick up senses from his mouth and his hearing.
Oh, that's
he can when he's walking towards the metal, he could taste the metal.
Right.
Or if he hears something far away, he could hear it, and we couldn't even hear it.
How was that? Right?
It was just traveling with him it was like I could taste metal. Where are we at? And, well, we're getting closer to train tracks or a fence.
When you're around a train track, do you taste metal?
No, I can't.
Can you smell it?
I could probably smell
diesel fuel.
Yeah, that's probably about it.
Huh. Yeah, okay, that's interesting.
So I thought I'd just put that in just with the vision, because once his vision closes,
right,
his other senses kicks in.
So, well, that's fascinating. So his vision, he was losing his rods. So he could still tell colors. He could read colors, but he was losing his peripheral vision. Okay, that's interesting. I wonder what that's called. There's a problem that people have where the middle of their eyesight goes away, and they can only see their peripheral vision. I'm trying to think what that is. They lose their ability to read because, well, that's in the middle of your field. A photon of light comes into your eye with select properties that we will perceive as colors. The hue is the wavelength of the light. Wavelength in the 400 to 500 range are very dark purples to blues. Wavelengths in the 500 to 600 range are greens to oranges with yellow in the middle. And wavelengths 600 to 700 range from bright reds to darkening to black. Since most colors detected as patterns of light are different wavelengths, Brightness refers to the amount of white light from the object. Saturation has to do with the amount of light of a select wavelength. If all the photons reflecting from an object were at 510 nanometers, the object would be highly saturated as green. I had a student who was a meth head, and she had, well, she was a meth head, and so she had meth head. But when I had her in class, even though she was clean, she couldn't tolerate bright colors. So when I put a lecture together, I had to put it on a dark background. And the letters couldn't be too bright. Otherwise, she'd get a headache and she couldn't watch the lecture, as interesting as that was. So, I don't know, it made her hypersensitive to bright lights. It's kind of interesting. The eye acts like a camera. The eye has a transparent, of course we don't have cameras anymore, we have telephones. The eye has a transparent sac called the lens that widens and narrows to focus light on the retina. Also leading to proper focusing is the cornea, or the curved outer cover of the eye that changes the light beam as it enters the eye. So the trick is to focus all of the light onto the back of the eye. And sometimes this doesn't happen. Sometimes your eye is too long, sometimes it's too short. And then we have either nearsightedness or farsightedness. The shape of the lens is controlled by a set of muscles around the lens called the ciliary muscles. When the lens is made to widen or shorten to allow for focusing, it is known as accommodation. And of course, if this is incorrect, then you won't be able to see very well. And this is known as an astigmatism. At birth, about 15% of males and 18% of females have severe enough eye problems to require correction. And I find this fascinating because as humans, we've had to survive for tens of thousands, maybe hundreds of thousands of years, probably millions of years. And if we had select individuals that had poor eyesight, how did they survive? Somebody must have fed them. At age 20, until presbyopia, and presbyopia starts about 40 sometimes, into your 50s, 48% of females and 32% of males require correction. This is half of females and 1/3 of males. So before we had glasses, how did these people survive? What did they do? They couldn't hunt. That's half of women. 1 out of every 2 women had bad eyesight to the extent that they couldn't see very well. 1/3 of men. So if you had a whole camp full of men, say 100 men, 33 of them couldn't see well enough to go out and hunt. Only 2/3 of them could go out and hunt. So did those 1/3 not make babies? Or maybe they stayed back and made the babies while all the other guys went out and hunted. I don't know. Maybe they became priests or something. I don't know. This is really weird to me. That we had so many people that were blind. I mean, not blind, but couldn't see properly. As odd as that may seem. Now the reality is that 2 out of my 3 wives have worn glasses, and they've worn glasses all their lives. So that might tell you something about my face. Only my first wife didn't need glasses. My son doesn't wear glasses. He's 46, and my daughter has started wearing glasses in her 30s. Good eyesight. We have good eyesight. I can remember when my mother started wearing glasses. And she started wearing reading glasses, and then she went to see the optometrist, and he gave her glasses. So she wore glasses for the rest of her life. My dad, on the other hand, wore glasses all of his life. Of my brothers and sisters, all of them wore glasses. My sister wore glasses all of her life. My oldest sister wore glasses all of her life. And the rest of my family didn't start wearing glasses until later. I only wear reading glasses. That's the reason I'm not walking around with glasses on. I can see much better without them. But I only wear reading glasses because I have presbyopia. But then again, I'm almost 70 years old. Nearsightedness or myopia is where the eyeball is too long and the clearest focus occurs before the light hits the retina. And that's nearsightedness or myopia. Farsightedness or hyperopia is where the eyeball is too short and the best focus point is past the retina. And astigmatism is a misshapen cornea. It's usually elongated that causes the light to pass incorrectly into the eye and cause the individual to be nearsighted. My wife, her last name was Urity. Her maiden name was Urity. So she always sat in the back of the room, but she couldn't see the board. And so for the her first grade and second grade, she couldn't see very well. And she was doing very poorly in school. But finally, I mean, they arranged it. This is in the South, so they arranged everybody in alphabetical order. Of course, she's a Urity, so she's way, way back in the back. And here she couldn't see the board. So my mother-in-law suggested to the teacher that she move her up. And of course, being the obsessive-compulsive people the teachers were in the South, they refused because it was alphabetical order. A's, B's, and C's get to be in the front. X, Y's, and Z's have to sit in the back. So they took her to the doctor and they got her glasses. And she said, you know, I used to see these trees and I could hear them and I could see the green, but I didn't realize there were individual leaves on the trees. She thought trees would be globs of green goop or something. And it's really kind of interesting. She still wears glasses.
I have a neighbor, a teenager. His eyes were misaligned.
Oh no!
Look forward. This one would look down this way.
Okay.
And his mom got him glasses slowly. Now there's like all in line. I guess.
So everything's okay.
Yeah, it's just it is.
It forced his eyes up.
Yeah. Yeah. So he positioned right correctly now. After he put his glasses on, they had a prescription where before that he had glasses, so he would look at you. This one would just wander off like this. Yeah. And it took a while to get used to it. And pretty soon he got glasses. I noticed that it lined up really nice, and his confidence is like way up there.
Oh, I can imagine.
He is like really talking. He comes by and before he was really shy. He would try not to. He knew he had the problem, but I think he was trying to find a way to connect. So it was like after the glasses,
people won't look you in the face.
Yes.
It bothers people to look somebody in the face if one of their eyes is looking over to the side. Mm-hmm. Yeah, it's bizarre. The iris is a muscular covering under the cornea that gives the eye its color. That's the iris right here. So you can see this person has, I don't know what color eyes they have. This is very confusing to me. I have blue eyes. Half of my family has blue eyes, the rest of my family has brown eyes. The pupil is an opening in the iris that allows light to enter the eye. The movement of the eyeball itself is controlled by the extraocular muscles. And of course, these are very important to your ability to see. One of the things that happens to you if you've had a brain, if you've had a concussion or something, a lot of times these extraocular muscles won't work properly, and your eyes are just kind of moving all over the place. And that's one of the things that we do if we're trying to figure out if somebody has a concussion. We look at their iris, and we try to determine if the pupil is at the right opening for the amount of light that's coming into their eyes. We watch their eye to see if it's moving erratically, and if it is, of course, that means that they've got a concussion. And of course, this is damage that could potentially become permanent if you're not careful. The photoreceptor cells of most animals are part of the retina on the back of the inside of the eyeball. The retina is very thin, but still contains 5 distinct layers. The first layer is the optic nerve fiber layer. This is working from the inside out, of course. The first layer is the optic layer. The second layer is the ganglion cells. The third layer are bipolar cells. The fourth layer is the actual photoreceptor cells themselves, the rods and the cones. And the fifth layer is made up of pigmented epithelium. Pigmented epithelium, so when you're driving around at night and a deer looks at you, the color of the color coming back to you, whether it's white or green or yellow, tells you how well they can see at night. The brighter the light showing back at you, the better that they can see at night. If you've ever taken a picture of a human being and their eyes came back red, it's because humans don't have very dark pigment. And this is one of the reasons why humans see so poorly at night. Now the interesting thing is that if somebody has rarely been outside at night, they don't go out in the dark, then a lot of times it will be kind of a dull red. But if it's an individual that spends a lot of time in the dark, especially without any light, they will have a brighter red color of their epithelium. So it all depends on how much stimulation they get. Now some people are afraid to go out in the dark at night, so they don't, and they never do because it scares them to go out in the dark. Other individuals go out in the dark all the time. I do. But then again, I see very well in the dark. My wife sees very poorly in the dark. My eyes will adapt relatively rapidly, just like my brother's did. Of course, he sees better in the dark than I do. But it's the fact that we practice. One of the things they used to do with my brother, they wouldn't let him go out in the sunlight. So they'd keep him in a darkened room or a darkened tent usually. Or they'd cover his head so that he would be able to see better in the dark. And then he'd go out and hunt at night, as ugly as that sounds. Or they'd send him into a spider hole. They dug tunnels all over Vietnam, and they had to figure out what was down there. So they'd send people in, and of course, sometimes they didn't come out. My brother always did, but sometimes they didn't come out. They usually attached a rope to them. Of course, that didn't really work very well because even if they'd attach it to one leg, and if somebody gets killed, they try to pull them out. Usually, the other leg gets stuck someplace, so they have to send somebody in after. At least they could follow the rope to figure out where he was. Anyway, that's the first time my brother ever went into a spider hole was in to get somebody that had been killed. The rods and the cones detect light that reflects off the pigment and epithelium. The rods and the cones release neurotransmitters that synapse with the bipolar cells. The bipolar cells synapse with the ganglion cells that form the optic nerve, and now we've got a picture. As you can see, the cones really are cone shaped, and the rods really are rod shaped. This is a rod, and that detects just detects light. The cones, of course, detect color. While most of the communication in the retina runs from outer layer to inner layer in a vertical pattern, the amacrine and the horizontal cells communicate with cells in a horizontal pattern. Now this is really important because this has to do with adaptation. So when you step out in the light if you've watched a matinee and you go out in the bright sunlight, one of the things that's going to happen is it's going to be blinding. Well, you need to adapt. Your eyes need to adapt. You certainly don't want to go blind just because you stepped out into the sunlight. And that's the job of the amacrine and the horizontal cells. They will shut everything down. Well, they will make you completely blind, but they will make it so that you're not reacting as negatively to the light. Horizontal cells connect receptor cells. Amacrine cells connect the bipolar and ganglion cells. And these allow for your light and dark adaptation. Rods and cones represent different functional systems The scotopic system consists of all rods in each eye, about 100 million cells, and this detects light. Now the amount of scotopic cells and the number of rods you have in your eyes have to do with how well you can see at night. So the question is, how important is it to see at night? And the answer is, well, if you're afraid to go out in the dark, then you probably can't see very well at night. Sometimes my dogs will bark at the front door. Sometimes there's people out there. Usually they just bark when somebody pulls up. I had 3 people stop at my house yesterday. 3 people. But everybody wanted to sell me something. Nobody wanted to borrow money, which was better. So I bought another load of wood. I've got 11 loads of wood at my house. So if you get cold, do you have a fireplace? Okay, well, don't worry about it. Anyway, yeah, I got 11 loads of wood. I just bought a 12th, so. Anyway, the irritating thing is that he's going to spend all of his money on a ceremony. His wife is in the hospital in Albuquerque. He's going to spend all of his money on the ceremony. He's not going to have any money to go back and forth. So, she's dying of diabetes. She's going to die. I'm sorry. She's going to die, and there's not a whole lot you can do about it. The ceremony is just to make her feel better, I guess. Anyway, he's using all the money to pay for the ceremony. I think he'd be better off just going with this here. But that's none of my business. The scotopic, sure it is, it's my business. I gave him the money. The scotopic system is very sensitive to weak light but cannot detect color. Light is captured in the scotopic system by the photopigment rhodopsin. The scotopic system is located primarily outside the fovea. So the more rods you have in your eyes and the more you use these rods, the better your eyesight at night. And some people have a lot more than 100 million rods, and some people have fewer than 100 million rods. So it really all depends. So here I've got my brother. He can adapt to darkness very, very rapidly, and it's because he has tunnel vision. I'm much slower at adapting, and probably I don't see as well as he does at night. But it's kind of interesting. We probably both have lots and lots of more rods than most people do. The photopic system consists of all the cones in the eye, about 4 million. Most of them, of course, are in the middle of the visual field. This system detects color and requires relatively strong stimulation, and therefore is primarily a day vision system. The photopic system is located primarily in the fovea, but is present in other areas of the retina, but in lower concentrations. So we can still detect color. You can detect color probably out to here, I'm guessing. But I told you about the red light that I kept seeing as yellow light. There are 3 forms of photopigments called opsins that detect the 3 color groupings, and there are 3 color groupings. The photopigment for scotopic system is rhodopsin, as I said before. The photopigment for the photopic system is one of the 3 configurations of opsins. All the photopigments in the eye are made up of 2 parts, retinol and opsin. Retinol is short for vitamin A aldehyde or retinaldehyde, shortened to retinol. And this is one of the reasons why you need to make sure that you take in adequate vitamins, because you need to, that's why they tell you to eat carrots. For good eyesight, you need carrots. Geez, I haven't had a carrot in a long time. But I'm not losing my color vision, that's for sure. But you need to take in all of these vitamins in order to be able to see properly. So one of the problems you have if you're an alcoholic is that you drink your meals, and you can live off of alcohol. I mean, it breaks down into sugar. So potentially you can live off of alcohol, And if you don't drink, any beer, you just drink the hard stuff, pretty soon your vision is going to is going to be affected because you're not taking any vitamins. And it's going to get affected. Other things are going to get affected, of course. Well, one of the things is your vision is going to go bad. So, because you're not taking in any vitamins. As much fun as that is. Light activates the rhodopsin molecule to rapidly lose its retinol, leaving an enzymatic receptor site on the opsin molecule. The enzymatic opsin molecule combines rapidly with G protein transducin. Transducin in turn acts on phosphodiesterase to convert cyclic guanosine monophosphate, cyclic GMP, to 5-GMP. You can see how slow this process is. Cyclic GMP inhibits sodium channels closing, and with the conversion, the sodium channels close. The loss of sodium ions depolarizes the neurons, and now we see what we see. Since we are dealing with millions of receptor cells at the same time, weak light will only hyperpolarize some of the receptors, and bright light will hyperpolarize most of the receptors. Thus, the response will always be graduated as the amount of light is detected. This form of response leads to sensitivity. It allows for adaptation. It allows the individual to integrate the stimulus over time. There are two types of bipolar cells that transmit information: off-center bipolar cells and on-center bipolar cells. On-center bipolar cells are activated when light is turned on. Off-center bipolar cells are activated when light is turned off, so that the eye still has visual capabilities. It detects light coming in from the periphery of the visual field. Photoreceptor cells release, and this is one of the, we're talking about glutamine. This is the reason that you get an after image. You get an after image because of the off-center and the on-center bipolar cells. Photoreceptor cells release the neurotransmitter glutamate. Glutamate is decreased in off-center bipolar cells when light is weak or turned off. Glutamate is decreased in on-center bipolar cells when light is turned on in the middle of the visual field. Okay. Each cone in the fovea, and this is where we get our after image. We get our after image from our off-center bipolar cell. Each cone in the fovea is connected to 4 types of bipolar cells: one on-center midget bipolar cell, one off-center midget bipolar cell, several on-center diffuse bipolar cells, and several off-center diffuse bipolar cells. Each midget bipolar cell connects to just one cone in the fovea. Each diffuse bipolar cell connects to several cones. When light is turned on-center bipolar cells depolarize on-center ganglion cells. When light is turned off-center bipolar cells depolarize off-center ganglion cells. Stimulus only affects the part of the retina that is in the visual field. The retina will create a mosaic of stimulation that the brain puts together as a picture. And for that reason, when you're looking at something, you can tell what color it is. You have a mosaic of what you're looking at. Remember, you've got a picture of at least 4 million cones telling us what color something is. There are various ways that the eye adjusts to different light intensities. The pupil of the eye will shrink with bright light and widen with dim light. Rods and cones have different thresholds. This is known as range fractionation. Photoreceptors will adjust their range of reaction depending on the intensity of the light. This will give the individual visual acuity despite the quality of the illumination, and that is where we need to stop. We are